And in fact, I got 263 references suggesting that, in fact, this is that there is something to be said for the comorbidity. Given that I now have some candidate genes, I looked for depression and MTHFR, ethanol and MTHFR, and the comorbidity, and I found some references. I found better information for TNF on depression, ethanol, and the comorbidity. So of course this is not proof, but it is consistent with the a genetic effect on this comorbidity, or at least on each of the separate phenotypes. And given that suggestion of the genetic effect on the comorbidity, I tested the positive predictive value of the queries against PubMed. And I should mention that in every case, and including the previous slide, the queries that I made are qualified by medical subject headings. And in this case, for the 263 citations for depression and ethanol, the actual query was this longer, more sophisticated query using mesh limitations on the query. So of those 263 citations, the question is, how many of them are really about depression and alcohol use disorders and potentially the comorbidity? I was able to check 179 of the 263 citations. Generally speaking, the ones that I could not check were written in a different language or are very old publications that are no longer available, at least through the University of Michigan Library. But of the 179 publications that were available, 171 of them I was able to verify that really do refer to both alcohol use disorder and depression, and which gives us a better than 95% positive predictive value for the query. So these results from these queries are quite believable. Not always accurate, but quite believable. So at this point, the PubMed evidence that we find is consistent with what we saw in Entree Gene, and we're starting to believe that there could be a genetic influence on this comorbidity. And in fact, this is consistent with the statistically significant association that was found by Hassan and Glick in 1993. I mentioned that I used medical subject heading qualifications on all these queries, so I think it's only fair to explain that there are some limitations to using mesh qualifications. First off, mesh is not an exhaustive annotation of all biomedical contexts, so some actual biomedical information will be missed. Equally, the annotation can be ambiguous. For example, in the query that I showed you a moment ago, I used depressive disorder mesh, which is one medical subject heading, or depression, which is a different medical subject heading. And it begs the question, which is the correct one? And the answer is that that's not obvious. So the annotation may be somewhat ambiguous. Also, in the query that I make, there's room for some ambiguity. And it's important to note that sometimes when a paper is published, the association, for instance, in this case between alcohol use disorder and depression, is found to be negative. So the number of papers that get published with negative results is relatively small, but it is something that has to be considered. Also, a lack of citations is not evidence of a lack of association. Equally, the co-occurrence of queried terms, which is what we're talking about here, is not proof of association. However, it does provide a quantitative measure of peer-reviewed research on the association. So in this case, we found 263 references on ethanol and depression, and that is in fact consistent with the statistically rigorous association found in a previous study. So at this point, we started off with some evidence of comorbidity. We identified a pair of candidate genes that show relatively useful evidence that they may play in this comorbidity. If we have more than one gene that plays in this comorbidity, it begs the question, do they interact in some way? And in this case, we used an algorithm locally developed at NCIBI called PDG-ACE. I'll explain that to you. PDG-ACE also gives us a hint at an environmental interaction with these genes. And in a moment, I'll show you the modeling as well. So PDG-ACE is an acronym for Prioritizing Disease Genes by Analysis of Common Elements. And the ingoing assumption for PDG-ACE is, like we have been talking about for complex diseases, that complex diseases are influenced by multiple genetic factors. And if, in fact, two different genes or two different loci are influencing a single phenotype, then there may be some common element of the genetic influences at those two different loci. You may be able to identify those common influences via gene ontology annotation or pathways annotations, and in some cases that works. However, that annotation is largely incomplete. So PDG-ACE uses natural language processing to bypass that limitation by screening through the text of entree gene records, and it generates novel, statistically significant, biologically plausible hypotheses on disease etiology. And this paper was published through AMIA, and it also has been accepted in BMC bioinformatics. It will be coming out this summer. So here's a graphical view of what PDG-ACE does. So we have two loci, chromosome A, chromosome B. Each locus can be a single SNP, a gene, a microsatellite marker, a cytogenetic band. The locus is relatively broadly defined. In this case, we're talking about TNF and MTHFR. So the text at entree gene that describes locus A, in this case TNF, is parsed. The text at locus B, in this case MTHFR, is parsed. And the parsed text is compared to a controlled vocabulary of biomedical keywords. And I've included a couple possibilities. Prefrontal cortex can be a biomedical keyword that might be important in neurological or psychiatric disorders. We actually developed three different controlled vocabularies. The first one has 2,531 keywords in it, and it's derived from medical subject headings. By parsing the medical subject headings, creating snippets of text that are likely to show up in the description of genes, and then eliminating the very common ones and eliminating the unique ones, so that the keywords that are left in the vocabulary are likely to show up and be useful in understanding disease etiology, and also be relatively rare. And the reason that you would want it to be rare is you can imagine that if you picked any locus A and locus B and one of your keywords was protein, it would be very common. So it would not provide information on the disease etiology. However, prefrontal cortex is a relatively rare term, and that might very well provide some information that would be useful. So we ran PDG-ACE. 10 million iterations. I'm sorry, I didn't mention the statistical significance. In assessing the rarity of a combination of genetic influences, we use permutation testing, comparing the initial observed locus to many, many random locus pairs, and score each keyword and rank that keyword in the distribution of the random scores, and if the score is sufficiently high, then we consider it to be statistically significant. And